Yeah, hello. Good evening, everyone. Am I audible to you? Sound is coming? Yes. Right, let's start. So, today we are starting with centrosome. Okay, ji? So, if we talk about centrosome, it is a non-membranous cell organelle. Right? Non-membranous cell organelles. If you talk about centrosome, it is present in animal cell as well as in lower plants. Where centriole is absent? Absent in higher plant. Or where centrosome is absent? Absent in higher plant. Right? Now we are going to discuss about the structure of the centrosome. Have a look. If you talk about the structure of a centrosome is consists of two centriole which is perpendicular to each other. This is one centriole and this is another centriole. So both centriole are present at which angle? 90 degree. And it is surrounded by pericentriolar material. This is not a membrane. This is what basically? Who is surrounding the centriole? It is pericentriolar material. And it is amorphous. Amorphous pericentriolar material. Amorphous means you can say about thick granular layer present around the which one? Both centriole. And both centriole are perpendicular. So this is the structure of a centrosome. Centrosome have how many centriole? Two centriole. And at 90 degree. They are present at 90 degree of each other. Is it clear? Understanding the idea? Got the things? Now we are going to deal about centriole. We are going to deal about centriole as well as we will discuss about basal body. Are you aware about basal body? Which give rise to cilia and flagella, right? So now we are going to deal about centriole and basal body. Centriole. In one centrosome, how many centriole is present? Two centriole. You just have a look. Student confused sometime. Centrosome, centriole. Centrosome is a cell organelle and it is made up of two centriole surrounded by amorphous pericentriolar material. It is not a membrane. Getting the idea? Now I am going to talk about which one? Centriole as well as basal body. So look here. Here is centriole and here I am writing basal body. So first of all, we will discuss about some differences between centriole and basal body, right? Then we will talk about their ultrastructure. Actually, centriole and basal body have same ultrastructure. But beside of that, their location, their number are different. So if you talk about centriole, its number is how many? 2 in a centrosome. In one centrosome, how many centriole is there? 2 centriole is there. Basal body are many in a cell. Number of basal body are what? Many. If you talk about centriole position, location, where centriole is present? Near the nucleus. Near the nucleus. If you talk about basal body, below the plasma membrane. Below the plasma membrane. You just have a look, I am drawing a cell. So in cell, this is the nucleus. So near the nucleus, you will find who is there? Centrosome is there. So suppose that it is centrosome, not a membranous structure. It is what basically? One centriole, and it is what? Another centriole, surrounded by amorphous layer. So it is centriole. Now if you talk about basal body, this is plasma membrane, and here who is there? This is plasma membrane, and this one is flagella. So in the basal part of the plasma membrane, who is there? It is basal body. Basal bodies. So number of basal body is equal to what? Number of cilia and flagella. Number of basal body is equal to what? Number of cilia and flagella. Getting the idea? Understanding the idea? Clear in the mind? But centriole, in one centrosome, how many is there? 2 is there. So you got the idea, understand the idea how centriole and basal body are different from each other. Right? This is basal body, this is centriole. Now we are going to deal about ultra structure of centriole and basal body. So what you have to know, both centriole and basal body have same ultra structure. Both centriole and basal body have same internal structure. Same internal structure. So now we are going to deal about the internal structure of centriole and basal body. Got the idea? We are going to deal about what? Internal structure of centriole and basal body. Have a look. So I am drawing the diagram. So look here, centriole and basal body internal structure. So if you look about the internal structure of centriole and basal body, it have cartwheel-like structure. Cartwheel-like structure. So cartwheel-like structure is present in which one? Centriole and basal body. And how it look in a structure? Here 9 plus 3 arrangement, 9 plus 0 arrangement is present. 9 plus 0 arrangement is present in both centriole and basal body. So 9 is what? Peripheral triplet. Here not a doublet, unlike cilia and flagella. Getting the things or not? And it is 0 means no microtubule. So only peripheral microtubule is there, in the center no microtubule is there. So peripheral triplet, 3 microtubules are present like this. This is one microtubule. With one microtubule, it is second microtubule connected like this. With second microtubule, third microtubule connected like this. This is A tubule, this is B tubule, this is C tubule. And this is called peripheral triplet. Got the idea? So how the overall structure of a centriole appear, let me tell you. Have a look. Give your attention. See, so I just draw this line for an imaginary line I drawn here. So triplet I am writing. So it is one triplet, it is another triplet, it is another triplet, it is another triplet, it is another triplet. How many? 1, 2, 3, 4, 5. Now it is another triplet, 6. Another triplet, 7. Another triplet, 8. Another triplet, 9. So these are 9 peripheral triplet. 9 peripheral triplet. I am removing these white dots. I just take it as a reference. Okay, so these are the 9 peripheral triplet. Now what happened? In center, there is a central hub is present. It is central hub. It is made up of protein. It is made up of protein. It is a central hub. Got the idea? It is proteinaceous, not made up of microtubule. Now, what the name of this microtubular triplet is there? I already told you, na? Here, who is there? A, B, C tubule. A, B, C tubule. Now, how they join together? Let me tell you how they join together. See the things. Here, A will join with C with a protein, AC linker protein, AC linker protein, AC linker protein, AC, 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 and AC linker protein. Got the idea? So they join by which one? A and C linker protein. So it is called AC linker protein. AC linker protein is present here. Getting the idea? Understanding the idea? So each triplet joined with the help of AC linker protein. Now what you will report? Below the X, below the A, there is a dense material present. Like this. With every, below every A, dense material present. Like this. Dense material present like this. With every, below every A, there is a dense material. And these material, dense material connected with A. Connected with A. Got the idea? So these dense material have a name. It is called X bodies. These dense material is called X bodies. Now there is another dense material present between A and B. Which one? X body. So this is another body. It is called bi bodies. So between two X bodies, there is another granular deposition is there. It is called bi bodies. And these bi body attach with AC linker. These bi bodies attach with which one? AC linker. So I attach here with AC linker. And this is called which body? Y bodies. Y bodies. Now what happened? X body and Y body are interconnected. Who is interconnected? X body and Y body. It is XY, XY, XY. Each XY and Y are interconnected. X body and Y body are what? Interconnected. Make it somehow longer. It will come up to here, right now. So it is interconnected. Got the idea? Understand the idea? Clear in the mind? Now from X body, who is arising? Radial spokes are arising from here. So 9 radial spokes are there. 2, 3, 4, 5, 6, 7, 8, 9. And this is what? Radial spoke. Radial spoke. Right. And this is hub. This is hub. This hub is made up of protein. And this is the ultra structure of a centriole and basal bodies. Got the idea? These are the ultra structure of which one? Centriole and basal body. Both have the same internal structure. It is looking like a cartwheel. It is appearing like a cartwheel, na? Is it clear? Understand? So now I am just writing one more line. Centriole forms the basal body of cilia and flagella. And centriole also give rise to a spindle fiber. So it is the function of centriole I wrote. So one, it is forming the basal body and it also give rise to which one? It give rise to a spindle fiber. So who is over? Centrosome is over. You got the idea? You got the idea about the structure of centrosome? Is it all right? It's done. Okay, ji, can we move ahead? Right. So centriole is over. So I am asking a small question here. In one centriole, how many microtubules are present? Answer it fast. In one centriole, how many microtubules are present? I am expecting the answer. You will answer it. Yes, children, carry on. It is easy, na? This is a structure of a centriole. And microtubule is what? This triplet is what? Made up of 3 microtubule. So in one triplet, 3 microtubule. How many triplet are there? 9 triplet. 9 into 3 equal to 27. You got the idea? So how many microtubules is present in one centriole? 27 microtubule. In one centrosome, how many microtubules is there? Answer it. In one centrosome, because in one centrosome have 2 centriole. One centriole have 27. So 2 centriole have what? 2 into 27. How much? 54. Very good. You got the idea? You understand the idea? So this is about the ultra structure of centriole. Not visible. What's happened? I think network may be an issue for you. Is it clear? So one centriole have 27 microtubule, one centrosome have 54 microtubule. Clear? Over. What is the function of centriole? Centriole form the basal body of cilia and flagella. Another function, centriole also give rise to which one? A spindle fiber. Done? Centriole or centrosome over. Now we are going to deal about microbodies. Then we will come on nucleus. So first we are going to deal about microbody. Microbodies. In the last line of the chapter, it is written in cell structure. After nucleus, it is written in NCERT, 2 line. Right, so what is microbody? So it is a minute, single membrane bounded, a spherical vesicle contain enzymes. Right. Microbody present in both plant and animal cell. It present in both plant and animal cell. If you talk about the size of the microbody, so diameter is 0.2 to 1.5 micrometer range. This is the size of the microbody. It is formed by endoplasmic reticulum. Who is forming this microbody? Endoplasmic reticulum forming this microbody. Getting the idea? ER is forming this microbody. How it look in a structure? So you will find there is a spherical membranous vesicle and it contain what? Enzymes. So it is single membrane, and here it is enzymes present. Okay, now I am giving a brief idea about type of microbody. I will not say much because NCERT said only 2 lines. So, but I will tell you the name, at least, and one function at least. So, example of microbody I am just writing. Example, 3 example of microbody we are going to discuss. One example I am just writing here is a spherosome. Another very common and popular, you know about it, peroxisome. And the third one I am just writing is glyoxysome. So these are the examples of which one? Microbodies. Got the idea? Now if we talk about a spherosome, it present only in plant cell. This is not present in animal cell. Getting the things or not? And function, if I wrote one-liner function, I can wrote like synthesis and storage of lipid. Synthesis and storage of lipid done by a spherosome. Now peroxisome present in both plant and animal cell. Plant and animal cell. Got the idea? But I am writing the popular function of in plant. In animal also a lot of function. In animal it also associated with detoxification, but unnecessary you confuse. Because detoxification related function I told you done by whom? ER. Even excessive alcohol anybody going to drink. So peroxisome have a role in the detoxification. 25% of methanol, ethanol, people use ethanol just going to which one? Detoxify by which one? Peroxisome. Even peroxisome doing role in what? Many such kind of things sometimes we eat. For that there is no digestive enzyme present in our body. So for that digestion occur by which one? Peroxisome. Peroxisome also have a role in digestion of prostaglandin. Got the idea? Peroxisome also have a role in digestion of long chain fatty acid. But that much I am not going to write here. I am simply writing here what? Peroxisome have a role in photorespiration. Because photorespiration written in NCERT, na? So peroxisome have role in which one? Photorespiration. Getting the idea? Otherwise, sometimes what's happened? Excessive teaching create a what? Confusion. Because many function overlap and you just get stuck. Here what to do? Sir told detoxification also done by peroxisome and ACR. What to do? Now what to choose? Getting the things? Unnecessary you will confuse. Right? Now I am telling about what? Glyoxysome. Glyoxysome present only in plant cell. Plant cell. And it contain enzyme. It contain enzyme for oxidation of fatty acid. fatty acid. Now in peroxisome, I also want to add one more enzyme. Peroxisome contain peroxidase and catalase enzyme. So if someone want to torture you, they may ask. If they don't want to torture you, they don't ask. Getting the idea or not? So peroxisome may contain enzyme which one? Peroxidase and catalase. So this is about the example of microbody, spherosome, peroxisome and glyoxysome. And one function at least I told. NCERT not wrote about all these. Done? Over. Can we move ahead? Right, G. So microbody is also over. Now we are going to deal about nucleus. So today our agenda is what? Finishing of this chapter. Done? So we are starting with what? Nucleus. Can we start? Can we move? Proceed? Okay, Ji. So have a look about nucleus. You know about discovered by whom? I already told who discovered the nucleus. Robert Brown. Earlier told in the history itself, na? Right? In which year nucleus is discovered by Robert Brown? Tell me, NCERT wrote. I'm expecting, yeah, in 1831. Done by whom? Robert Brown. Good one. Now we are moving ahead. Have a look here. So nucleus, it is a double membrane bounded a structure present in almost all eukaryotic cell. Almost all means not all. Some exceptions are there. Getting the things or not? Almost all means most of the eukaryotic cell. I just use this word to make you understand. Almost all doesn't means all. Able to understand? Right? So what is happening basically? Most of the cell have a nucleus. Most of eukaryotic cell contain how many nucleus? One nucleus. But certain exceptions are there. Exceptions are there. So I am writing about exceptions related to nucleus. And what is the exception? Exception, I am writing cells without nucleus. Which one? Mature mammalian RBC. RBC. And another example I am writing, sieve tube element. Sieve tube element. What is there? Nucleus absent. Nucleus absent. Got the idea? In mature mammalian RBC and sieve tube element, nucleus is absent. Now I am giving you some another exception where more than one nucleus is present. So another exception I am writing in paramecium. How many nucleus is there? Two nucleus. Two nucleus is there. Another example I am writing in tapetum. Tapetum is also what? Bi or multinucleated. So these are the exceptional conditions where nucleus number is either absent or it is more than one. Are you getting the idea? So these are the certain exceptional example I wrote. Otherwise, Most of the cell have how many nucleus? One nucleus. Is it clear? But it is in eukaryotic cell. Mind this line. Not in prokaryotic cell. We already discussed in prokaryotic cell well-defined nucleus is absent. Now we are going to deal about the structure of nucleus. Done? So now we are going to deal about the structure of the nucleus. Okay, Ji, have a look here. See the things. So I'm just drawing a cell. This is a cell. And in this cell, this is what? Nuclear membrane. Actually, for nuclear pore, I did like this. Otherwise, it becomes difficult to erase here. And that is the reason I did like this. So this is outer membrane, inner membrane, this is outer membrane. Both are parallelly present. You see? And these membranes fuse at certain interval. Agree or not? These membranes fuse at certain interval. So these membrane is not a continuous structure. Right? So these are nuclear membrane, and inside the nuclear membrane, who is there? Fluid filled matrix is there. This fluid filled matrix is called nucleoplasm. And outside of the nuclear membrane, what is there? It is cytoplasm. And cytoplasm also have fluid. Here it is also what is there? Cytoplasm. Here also who is present? It is cytoplasm. Fluid filled matrix is in cytoplasm is cytosol. So here it is cytoplasm. Are you getting the idea? So fluid in the nucleus is different and fluid in the cytoplasm is different. Cytoplasm present outside the nuclear membrane and inside of which one? Plasma membrane. And you better know, na? You already know how many times we have to say. Outer membrane of the nucleus is continuous with which one? Endoplasmic reticulum. It is continuous with endoplasmic reticulum. You got the idea? Right. And also it contain, when it continuous with endoplasmic reticulum, who is also there? A ribosome also present on the outer membrane. So outer membrane of nucleus also contain which one? Ribosome over it. So this is which one? Outer membrane of nucleus. OM in short I am writing. And inner it is IN, inner membrane. And you better know outer membrane contain ribosome. Contain ribosome. Are you getting the idea or not? Contain ribosome. Okay. Now there is a space between two membrane. Is there na? That is perinuclear space. Perinuclear space is present between the two nuclear membrane. And what is the diameter thickness here? It is 10 to how much? 50 nanometer in diameter thickness. How much? 10 to 50 nanometer. It is perinuclear space. Now, nuclear membrane, I already told it is not continuous. So wherever nuclear membrane is not present, that area is called nuclear pore. Nuclear pore. And don't confuse, this nuclear pore is not a open structure always. It is guarded structure. So it is guarded by annulus. Who is guarding the nuclear pore? Annulus is guarding the nuclear pore. Means through nuclear pore, entry and exits are restricted. It's not like anybody can enter, anybody can out. When guard is there, guard will check na who have to go in, who have to not go in. Who will go in, who will pass and who will not pass. Understand the idea? So it is guarded. It is which one? Guarded by annulus. Is it clear in the mind? So this is nuclear pore. What these nuclear pore facilitate? So nuclear pore facilitate the transport of RNA and protein in both direction. Both direction means from cytoplasm to nucleus and from nucleoplasm and from nucleoplasm to which one? Cytoplasm. So it facilitates the which one? Transport. Getting the things? You better know, na? Transcription. Where transcription occur? Occur in nucleus. Translation occur in cytoplasm. So for translation, ribosome go out or not? Go out. So ribosome going out from nuclear pore, from inside the cell to which one? Outside the cell. Sorry, outside the cell I am telling. Inside the nucleus to which one? Cytoplasm. And now, ribosome formation occur in nucleolus. I will tell about it. So for nucleolus, ribosome have how many components? RNA and protein both. So they will also enter in which one? Nucleus, na? You are getting the idea? For formation of ribosome, even protein go inside, na? Protein go inside, otherwise how the ribosome will form? So ribosome and protein moving in which one? Both the direction. Through which one? These nuclear pore. Okay, anyhow, now in nucleoplasm, what we are finding, there is a dense material present here. Getting the things or not? Its number is maybe one or more than one. Able to understand? And this structure is called nucleolus. Nucleolus. So its number is one or more than one. So its number is not one only, it may be one or more than one. And it is non-membranous structure. Non-membranous means it is in contact with which one? Nucleoplasm. It is, if membrane is not there, it will not separate, na? So it is in close contact of nucleoplasm. Of which one? Nucleoplasm. Non-membranous structure is there. No any membranous are separating nucleoplasm and nucleolus. Getting the idea or not? And what is the function these nucleolus is serving? These nucleolus are serving the function of, it is the active site of synthesis of ribosomal RNA. Ribosomal RNA, function number one. And function number two, it is also called ribosome factory. Ribosome factory means ribosome formation occur here. When rRNA and protein assemble together, what happened? Ribosome formation occur. So these are the two important functions served by these nucleolus. Are you getting the idea? So these are the dense material present in which one? Nucleoplasm. Now inside the nucleoplasm, there is, and especially if we are talking about nucleus, nucleoplasm, we are talking about which nucleus? Nucleus in interphase stage. So we are discussing here the structure of a which nucleus? Interphasic nucleus. Keep in your mind. Interphasic nucleus. Because nucleus is not present every time in the cell. Nucleus is present in interphase. Nucleus disappear in which one? Degraded in which one? Late prophase. Not present in metaphase. Not present in anaphase. Further regenerate in which one? Telophase. This picture is there in your mind or not? So what are the structure is there? We are talking in the reference of a interphasic nucleus. So in interphasic nucleus, what you will find? You will find there is a network of, getting the things or not? Thread-like network of which one? Nucleoprotein. And it is called which one? Chromatin. Getting the things or not? So this is what? This is what? Chromatin. I am writing here chromatin. Chromatin. Got the idea? What is this chromatin? What is this chromatin? Let me write about this chromatin here. So it is thread-like network of nucleoprotein. Nucleoprotein means what? Here two component is there. Nucleic acid is there and who is there? Protein is there. So this is a thread-like network of nucleoprotein present in interphasic nucleus. Present in interphasic nucleus. So who just this chromatin name, who given it? Given by which one? Fleming. Fleming. Getting the idea or not? Who have given the name? Fleming have given the name. Is it clear? Is it clear? Right? So what he did basically, we stain the nuclear material inside the nucleus with the help of a which one? Basic dye. So these chromatin become visible after staining with which one? After, let me change the color. After staining with which one? Basic dye. Basic dye, we find which one? These chromatin. And this network called chromatin by which one? Fleming. And what is this network? It is a thread-like network of nucleoprotein present in the interphasic nucleus. Composition of chromatin. Now I am telling you composition of chromatin. Composition. So what NCERT wrote in the composition? Mainly made up of DNA and histone protein. Mainly made up of which one? DNA and histone protein. And who is also present? A small amount of RNA and non-histone protein also present. Who is also present? RNA and a small amount of non-histone protein also present. So if you talk about composition, how many things are there? DNA, RNA, histone protein, non-histone protein. So these are the compositions of which one? Composition of which one? Chromatin. Understanding the idea? Chromatin is present in which stage? Interphasic nucleus. Where it is present? Interphasic nucleus. It is not present in cell undergoing the progression of division phase. When cell dividing, chromatin converted into which one? Chromosome. Able to understand? In late prophase, in metaphase, in anaphase, it become what? Chromosome. So what happened? When chromatin condense, during division phase, it is called chromosome. Chromosome. Got the idea? Is it clear in the mind? So when chromatin condense during division phase means prophase, metaphase, anaphase, it is called what? Chromosome. In telophase, once again it become what? Chromatin. Because you have studied now, telophase is reverse of prophase. In prophase condensation occur, in telophase decondensation occur. Is it clear in the mind? Now I am asking a small question from you. And it is a small common sense based question. Try to answer it. Okay, Ji? Can we proceed? Have a look. I am asking a small question. Try to answer. So suppose that it is a human somatic cell. In which stage it is there? G1 stage. And here chromatin is there. First I am asking a question to you. In G1 stage of the human somatic cell, how many chromatin is present? How many chromatin is present? In G1 stage. Tell me. Answer it. So the answer of the question is what? 23 pair, or you can say 46. In one line you can write what? 46. How many chromatin is there? 46 chromatin is there. So you can say, sir, 46 chromosome, look here, it is nucleus. So chromatin present like this, 1, 2, 3, 4, 5, 6, 7, and it is forming a network like a structure. How many are total? How many are total? 46. You know na, in your somatic cell 46 chromosome is there. So who is converted into chromosome? This chromatin is converted into chromosome na. These chromatin condense and then form chromosome. So 46 chromatin condense and form how many chromosome? 46 chromosome. Are you getting the idea? Is it clear? Came in mind or not? Why I told 46? You are telling the 46 chromosome. So 46 chromosome coming from what? Chromatin na. Chromatin condense and who form? Chromosome form. So number of chromatin and chromosome are same na. Is it clear? Understand the idea? Right? Now I am asking, in one chromatin how many DNA is there? How many DNA? Answer this question. Very good answer. In one chromatin, one DNA is present. In one chromatin, one DNA is present. So in 46 chromatin in your cell, how many DNA is there? In human somatic cell, how many DNA is present at interphase, is present at G1 stage? Answer of the question is how much? 46. Are you understanding how 46? 46 DNA. Because 46 chromatin is there. Each chromatin have one DNA. 46 chromatin have how many DNA? 46 DNA. Now NCERT wrote a line. Ki when you arrange every DNA in line, all 46 DNA in line, just one after other. I'm just telling. So when the overall length will come for all the DNA is how much? Approx 2 meter. Approx 2 meter come. Actually it is 2.2 meter. But this line is written in genetics topic. Actually if you calculate, I will teach you after during biomolecule. Not in biomolecule, you can say during which one? Either molecular genetics. Actually it is how much meter? 2.2 meter, but approximately in cell topic it is written 2 meter. So when all the DNA, getting the things or not, are placed one after other in a line, how much total length create in case of a human? When we remove all the DNA from a human cell, it is approximately 2 meter, and exactly it is how much? 2.2 meter. Oh, got the idea? Understand the idea? It is exact. Done. Over. Clear? Now I am telling you about this somehow in better way. Have a look here. Have a look here. It will help us in cell cycle also, na? So do the homework here only. So in cell cycle, your concept will be better. Now look here. This is a what? What is this? Tell me, what is this? This is a DNA. How many DNA? How many DNA is this? One DNA. Here it is one DNA. Right? Double helically coiled structure. It is a long one DNA. Now, what is the difference between these DNA? How these DNA converted into a chromatin? So these DNA, just have a look here, wrapped around. I'm just giving a basic idea. Much detail we will deal during nucleosome model. So these DNA is wrapped. I'm just changing the color. Around which one? These DNA is wrapped around histone protein network structure. So when DNA wrapped around histone protein structure, some non-histone protein came, some RNA will came, and then it become chromatin. It become chromatin. How many chromatin? One chromatin. So one chromatin have how many DNA? One DNA. Are you getting the idea, understanding the idea? Now these chromatin undergoes in condensation. These chromatin undergoes in condensation, condense. And after condensing, it become forming a such kind. It become like this. It become like this. And this is called what? One chromosome. Or better to say, people confuse, it is called chromosome with single chromatin. Chromosome with single chromatin. You got the idea? It is, you can say it chromosome also, and you can say it chromatid also. Many times student confuse. No sir, it is not chromosome. It is chromatid, na? Because in your mind, what is the picture of chromosome? In everyone's mind, generally chromosome picture is like this. Got the idea? Chromosome picture is like this. And when you say this one as a chromosome, they say, well, how it is chromosome, sir? Here it is two chromatid. So here it's chromatid, na? We can also say it what? Chromosome with single chromatid. And this condition is called monad. This condition is called monad. Got the idea? Understanding the idea? Is it clear? So in this chromosome, you tell me how many DNA is there? How many DNA is there? One DNA is there. Now I am telling about this chromosome something. Wait. This is a invagination, na, constriction. Na? So this is called primary constriction. This primary constriction have another name, centromere. Done? Primary constriction is also called centromere. This is another constriction, it is called secondary constriction. And about secondary constriction, I am just writing one thing. It helped to identify the DNA, chromosome. It helped to identify the chromosome. Why? How? Because its position is fixed on a chromosome. So secondary constriction helping us to identify the chromosome because its position is fixed on chromosome. Suppose that 15 chromosome. So the position of secondary constriction is in a particular point. So if you find it is at that point, you can say it is what? Secondary constriction. Then you can say it is 15 chromosome. An example. Now, along with secondary constriction, here NOR is present. Nucleolar organizer region. And these NOR, full name is nucleolar organizer region. And these NOR form nucleolus. Nucleolus is formed by whom? Nucleolar organizer region. Are you getting the idea? Now, these are bulge out region. In NCERT it's shown upside, doesn't matter. This bulge out region above the secondary constriction is called satellite. Is called satellite. Got the idea? These bulge out region above the secondary constriction. What is satellite? Satellite is bulge out region above the secondary constriction. Satellite is bulge out region above secondary constriction. And these satellite not present in every chromosome. Don't confuse. Where this bulge out region is there, it is present. But at this standard, I will not teach in that much detail which chromosome have satellite is and which don't have satellite. Getting the idea or not? So you just find like bulge out region is called what? Satellite. Clear in the mind? Now I am telling the terminal end. How many terminal end is there in this case? Chromosome with single chromatid. 2, one this side, one this side. It is called telomere. It is called telomere. Here it is also which one? Telomere. Picture came in mind or not? Now if you tell about, so here how many telomere is there? 2 end, each end have a telomere. Keep in mind. So it is telomere. Let me erase meanwhile this diagram. Then I will come on this diagram later on. Wait for a moment. So it is what basically? Telomere. Clarity is coming or not coming? I am giving you some more information about telomere. Telomere rich in guanine. Telomere rich in which one? Guanine. Because DNA present here, na? So in telomeric region of DNA, in telomeric region of DNA, who is there? Guanine nitrogenous base is more. Getting the things or not? And telomere also play a role. It prevents 2 chromosomes to fuse together. It prevents 2 chromosomes to fuse together. If you cut the telomere, 2 chromosomes will fuse. Mutation occur, chromosomal aberration occur. You got the idea, understand the idea? And telomere, another interesting fact I am writing. Telomere, its size decrease with age. This standard I am not going to tell you how it decreases with cell. Otherwise a question came in mind, sir, with age it is decreasing, size of telomere. Size of telomere is size decrease with age. So we'll study it in somehow higher standard. It is not the right time to talk about it. Otherwise a lot of time will take. Got the idea? So telomere size decrease with age. So anybody can find the age of a person by removing the cell from a body. Based on what? What is the length of the telomere? Are you getting the idea, understanding the idea, clear in the mind? Okay. So we determine the age. Suppose that some very virtually burnt dead body came. So how to determine the age? What was the age of the people? So we are removing their DNA, na? DNA, na? Getting the things or not? And after that, we can determine the age in forensic study. You are getting the idea, understanding the idea. Is it clear? So, right. So about the telomere, you got the idea? Done? Now we are moving ahead. Have a look. Just see the things. So this is about the structure. Clear? Clear? Done? Okay, Ji. Now I am telling what is your perceptions. What is the perception of chromosome in your mind about that? Let me talk. Can we move ahead? Can we move ahead? Yeah. Have a look here. Now see, I am drawing once again this diagram. I am this time not drawing secondary constriction. This time drawing only which one? Centromere. Got the idea? Okay. Now look, what happened in S phase during DNA replication. In S phase during DNA, what happened? DNA replication occur. So when DNA replication occur, what it become? During S phase, this will happen. This will happen. This converted into this. DNA replication occur. So during S phase, what happening? Formation of sister chromatid. During S phase, what's happened? Formation of sister chromatid. Are you getting the idea? Understanding the idea? And this position is what? Centromere. At centromere, both sister chromatid attached. Right? And this is called, here you will find there is a disc-like substructure present in both sides. And this disc-like structure is called kinetochore. Right? And this is called centromere. Getting the thing? This condition is called monad. And this condition is called dyad. Got the idea? Understanding the idea? Clear in the mind? Is it all right? So, I am asking a question. Try to answer it. In dyad stage, how many chromosome is there? How many chromosome is this? One chromosome. Here also one chromosome. This is chromosome with single chromatid. This is chromosome with double chromatid. But still it is the same chromosome. Why it is the same chromosome? Because both are attached. Two unit, when it become? When it will separate, na? When it become, we can say two. When they detach from centromere also. When it detach from centromere also, then you can say two, na? Until unless it is not detaching, you can say two. I am asking a question to you. You will understand better. How many ball is this? One ball. How many ball is this? One ball. When it become two ball? When it becomes separate like this? Because still it is attached, fused together. It not have an independent existence. Until unless it not have an independent existence, you can't say two. You are getting the idea? So same story is here. One DNA another DNA. And always keep a thumb rule. Number of chromosome is always equal to number of centromere. Clear? You will never confuse. Number of chromosome is equal to number of centromere. Never a confusion occur in your mind. Got the idea? Is it clear? Now have a look, dear children. I am asking in dyad stage, how many chromosome? In this structure, 1. How many chromatid? 2. Very good. How many DNA? 2. Each chromatid have 1 DNA. Keep in mind. It will help you. Each chromatid have 1 DNA. In a chromosome, number of DNA is variable. In G1 phase, 1 chromosome have 1 DNA. In S phase, 1 chromosome have how many? 2 DNA. And better to say not technically chromosome, better to say chromatin. Getting the idea now? Because chromosome divide in which one? Form in which one? Prophase and metaphase. Anyhow, how many kinetochore is there? How many kinetochore? 2 kinetochore. A spindle fiber came from this side attached with kinetochore. Got the idea? How many telomere is there? How many telomere is there? Here 4 telomere is there. 1, 2, 3, 4. And assume I am drawing the diagram like NCERT. Suppose that here, look here. Suppose that here after some constriction, here bulge out region is there. So this constriction is called secondary constriction, and this bulge out region is called what? Satellite. This bulge out region is called what? Satellite. And that is the diagram in your mind. In your mind, chromosome picture is there, na? This picture, any differences between these two? Both are same, na? Both are same, na? Actually, it is like this. You just make a cartoon model like this. Able to understand, got the idea, understand the idea. So I hope it will give you a clarity. Got the idea? Done? Okay. So it will help you a lot in cell cycle to handle the numerical. Otherwise, students struggle if they don't have idea about these things. Clear? Done? So our effort will save in cell cycle. Can I expect? Okay, ji. Now we are going to talk about classification of chromosome based on position of centromere. Classification of chromosome based on position of centromere. It is damn easy, everyone know. I am writing. It is metacentric, submetacentric, acrocentric, and telocentric. Now I am writing about this. First I am writing about shape of all this. Shape. Shape we study in which one? Anaphase stage. I will tell you how. So metacentric have V shape. Submetacentric have L shape. And acrocentric have J shape. And telocentric have I shape. So V shape, L shape, J shape, and I shape. Now I am telling position of centromere. Position of centromere. Here it is in center, median. median. Arms, both are equal in length. Equal in length. Got the idea? Here it is submedian, away from center. One arm long, one short. One short arm, one long arm. And one long arm. One short arm, one long arm. Short arm is called P arm. Short arm is called P arm. I will draw in the diagram and I will tell, don't worry. Wait a minute. Then here what happened? J shape one. It is subterminal. Subterminal centromere. Not on tip, in subterminal region. And one arm very long, one very short. Very short, one very long. And telocentric, terminal centromere. How many arms is there? Only one arm. Got the idea? Now I am telling you the images, diagram. Look here. This is metacentric. In center, who is there? In center, centromere is there. In middle, centromere is there. Next one is submetacentric. You look here. Submetacentric. One arm is shorter, another arm is longer. It is submetacentric. It is short and it is short arm and it is long arm. And short arm have another name. It asked 3 years back in NEET, P arm. And long arm is called Q arm. Getting the idea? Shorter P, longer Q. Now submetacentric diagram I am drawing. Submetacentric. One arm very short, another arm very long. Here is centromere. Centromere. So one arm very short, another arm very long. Right? Short, long. Also P, Q. Now I am coming on telocentric. Telocentric have how many arms? Only one arms. You will find both arms is not present. Tip, centromere present on tip. Terminal, it is telocentric. Now NCERT diagram when you refer. Third diagram and fourth diagram. Their intention to say what? Metacentric, submetacentric, acrocentric, telocentric. But you will find in third and fourth, they are showing two arms. And that is the reason many times some teacher frame question, especially in these two states. Ki shorter arm, telocentric. It is not telocentric. Intention of NCERT author to teach you what? Telocentric through 4 diagram. One for meta, one for submeta, one for acro, one for telo. But how the people will trap you, teacher will trap you, they give you this diagram. Fourth one. And if you write telocentric as an answer, they will make it wrong. Why wrong, G? Because they will say, see, look, both arms is shown. Getting the thing? Their intention is telling you, teaching you telocentric. What I want to say, are you getting the idea or not? But anyhow, diagram must be more better. Diagram must be more better because they have seen both the arm there. Are you getting the idea, understanding the idea? Clarity came or not? So these are about the classification of chromosome based on what? Position of what? Centromere. Now we are going to talk about two more terminology. One is karyotype. So karyotyping is what basically? It is arrangement of chromosome based on their length, position of centromere, etc. Because I not told banding, so I am not writing, is called karyotyping. So when each and every chromosome have a picture, now when you click a picture of a chromosome, you will find na someone long, someone short, someone have which one? Centromere in middle position, someone have centromere in away from middle position. Are you finding the idea or not? And what is another term is there? Ideogram. Ideogram is what basically? Diagrammatic representation of karyotype. Diagrammatic representation of karyotype is called ideogram. When you draw the diagram of every pair of chromosome, so longest pair of chromosome we keep, I am just giving you an idea. Suppose that this is the longest chromosome. So I am giving longest pair of chromosome. So it is what? First chromosome. Who is the first chromosome? Longest chromosome is first chromosome. Usually in karyotyping, what we will do? Second longest chromosome, second longest pair. So usually we are arranging based on the size, all 23 pair. And we just assemble sex chromosome in the last. Sex chromosome in the last. Getting the idea? So this arrangement is called karyotyping. But you don't confuse yourself. According to Human Genome Project, story gets changed. Earlier we teach as a teacher what? Ki in karyotyping, when we arrange the chromosome, we arrange according to size. So the longest size is the first and the smallest in the last. We just told earlier like this. But now according to Human Genome Project, it is not true. Human Genome Project told a smallest chromosome in our body is 21st. Second smallest is? Second smallest chromosome is? Smallest chromosome. Second smallest is 22nd. But when you look the karyotype, in karyotype first we draw home. 21, then we draw home. We write keep home, 22nd. Means who is smaller is before, who is larger is after. So now according to Human Genome Project, that is not going to followed completely, able to understand. Otherwise earlier my teacher told like what? Kiji, look, arrangement according to decreasing in size. It is not everywhere applicable. Otherwise you will say who is the smallest? 22nd is the smallest. But actually who is the smallest? 21st chromosome is the smallest. Are you getting the idea? Understanding the idea? And in karyotyping, sex chromosome is placed in the last. Are you getting the idea? So when you are arranging, process of arrangement is called karyotyping. After arranging, when you are clicking a pic, it is called ideogram. You got the idea? Understand the idea? Is it done? Okay, Ji. Then thank you very much. Bye-bye. If any doubt is there, you are welcome with your query. You are welcome with your doubt. Right? If any problem, any doubt, hassle-free you can ask. And children remain motivated. It is most important things. Okay? Yeah. Biomolecule, the next chapter. We will start. Don't worry. Is it clear? Done? So, should we discontinue now? So, I am just sharing I forget to share always, sharing the which one? Screen to you. Biomolecule there in this week. Biomolecules, some one lecture topic is there in this week. Getting the idea? So this is you just scan it, board will come to you. Right? Biomolecule is there, up to amino acid it is there. Probably biomolecule is up to amino acid. In one lecture we will finish up to there, don't worry. Next lecture, 5 minutes I will teach you about giant chromosome. Because not in NCERT, so I will just give actually the line what is going to ask in exam, probably, and after that we will start which one? Biomolecule. Yeah? It's all right? It's okay, bye-bye. Good night to everyone. Bye.